David is here. Ta-da! Sort of works. Let's see if the rest of it works. First you turn it on. It complains. Yes, it works. Okay, very good. This is a meeting. I'm just trying to find places to put the various appendages with which I've been saddled here. Okay, does the microphone work, sort of? The room is small enough that it doesn't really matter, I'm sure. Okay, I guess I'm the beyond part of the title of the conference, and I guess to pick up on a word or two from Director speech, it's also, I think, about the how shall I put it, boundary-free nature of theoretical physics. It's not just national boundaries, but I think it's also intellectual boundaries that are, how shall I put it, in the best traditions of our field, we trample on intellectual boundaries as well as trample boundaries, international boundaries all the time. Okay, so my talk is going to be about something that will not be mentioned again during the next 3 days, namely it's a topic in biology, and I tell you roughly speaking what it is on the front here, but basically I'm presenting this story to David on his 75th as an example of how the theoretical physics way of thinking can do something useful for biology. We've talked about this from time to time, but I actually never really had the opportunity to tell him in detail about a particular application thereof. So it's kind of, I'm looking forward to this. For some reason, something seems to have happened here. There's, okay, okay. I think this is now correct. All right. So my thesis is that biology needs help from theoretical physics. Biological big data is, which you've no doubt heard about in the newspapers or reading Science or Nature, is creating a need for mathematical frameworks in order to comprehend, make use of the large quantities of data that biology is creating in many different directions. And these mathematical frameworks are similar to what theoretical physicists have already, have always been doing for the physical sciences. I don't have time to explore this in any depth, so I thought what I would do would take a look at a concrete example, and it's just something that one theoretical physicist who got interested in biology found it interesting to do, and you'll see there will be very little overlap at a direct mathematical level with the kind of stuff that you guys do every day, beautiful equations and so on. On the other hand, I think if you look at a meta level, you'll see that there's something going on in what I'll talk to you about that is worthy of the attention of theoretical physicists. Okay, so at a somewhat lower level, what I'd like to remind you is that biology, in biology, what matters in many cases is a probability distribution. So, when you look at a scene, some shower of spikes travels down the optic nerve and goes to the brain, and somehow when you look at a particular thing, like David sitting here, a particular shower of spikes will go down your optic nerve and your brain will transform it into a broadly smiling, couldn't possibly be 75-year-old gentleman who's looking pretty good, all things considered. And if I turn away and do it again, I'll get a somewhat different shower of spikes coming down the optic nerve, but it'll still be transformed into the same picture. So somehow David is a particular instance of, every time I look at him, it's a draw from a distribution on a very high dimensional space. And so it's a general problem in biology to figure out, well, how do you learn, how do you characterize these distributions? They're very important because it's the way you learn about the outside world. It's the way biological entities make decisions. They get many, many noisy inputs, and they have to decide, I am now in a state where it's really important to do something different from what I've been doing before. And, well, okay, before you can manipulate such things, you have to learn how to understand them, how to characterize them. So immune system diversity, I'm going to have to watch the clock, provides an instructive concrete example, and that's what I want to talk about today. Okay, so what do I mean by immune system diversity? Well, All right, so let me give you a crash course in adaptive immunity. All right, so your blood contains red blood cells and white blood cells, and actually if you start looking at what goes on in your blood, you will immediately get completely disgusted. There are so many different types of cells that it's hopeless. However, there are two main classes of what you would typically refer to as white blood cells that play a role in immunity, and these are T cells and B cells. Now I'm going to talk mainly about the genetic, so to say, the genomic content of T cells and B cells, so I have to tell you a little bit about what's going on. So you have all of your blood cells are made in the bone marrow. There are some number of stem cells, and on a regular basis they send out new proto-blood cells, and some of them become cells that perform jobs for the immune system, and these are primarily T cells and B cells. So each time a new cell is made, if it's going to be a T cell or B cell, eventually something called eventually the genome of that cell, the new cell that's produced, is edited in a somewhat random fashion to create a new thing that's going to develop into a T or B cell that has a unique gene, different from any that has ever been made before in your body, for a particular protein. It's the protein that gets expressed on the surface of the cell, and it's that surface receptor that gives a T cell or B cell some specificity in what it can see, what it can detect, what it can respond to. So new cells that are made in this way are tested against yourself, namely fragments of proteins that you make, and this proto-cell is going to be rejected if its recognition of you is too weak. That means it's probably not a very good T cell or B cell, or too strong, and that means when it gets loose in your body it might generate what amounts to autoimmune reactions. Okay, so these cells go into the periphery as naive cells. They wander around, and every now and then they will recognize something that shouldn't be there, a pathogen, and that then stimulates multiplication of, or expansion of the size of the clone of this particular type of immune cell, and with any luck it will then clear the pathogen from your body. Okay, I'm going to fuzz over the differences between the roles of T cells. They recognize foreign bodies that are inside your cells, and B cells, they're roughly engaged in detecting free-floating stuff like viruses or bacteria. But in many ways, they have so many similarities that from the point of view that I'm going to be talking about, we can smudge them together. The point of this genetic diversity, the fact that every new cell of this type has a different gene for this thing that gets expressed on the surface, the point of it is so that in the very large number of distinct immune cells that are in your body, some new pathogen that's going to attack you has some chance of being recognized by at least one of this standing army. So you have a standing army. Each member of the army has a different weapon. This weapon will only kill a particular enemy soldier. You have to hope that you have the right range of weapons available so that no matter which enemy soldier shows up inside your body, you have somebody there that can actually deal with it. And the whole point of my talk, or the thing I'm going to talk about primarily, is how to understand this genetic diversity, how to quantify this genetic diversity of immune cells. So how does it get made in the very first place? Well, it's quite a remarkable process. So it's called recombination, VDJ recombination. It works in a way that I call the Chinese restaurant menu approach. That is to say, you have a long, on some chromosome there's a long stretch of DNA. Column A is a series of possible choices for one piece that you're going to put together to make the final gene. Column B is another set of element modules, and column C is another set of modules. There are enzymes around that can do repair work on the chromosomes, on your DNA. Your DNA is being broken up all the time, and you have to have enzymes around that will repair mistakes, repair breaks. And some kind of some kind of process of evolution has led to the existence of a suite of enzymes that will do the following tasks when given the signal to go ahead and do it. They will, let's see if maybe I can point to this more precisely. Okay, let's focus on, so what's represented here is the end result. It's a receptor that is being expressed on the surface of a cell. The rest of the cell is completely invisible. There are tens of thousands of these guys on the surface of one of these cells. It consists of two proteins, an alpha chain and a beta chain. I'm going to focus on the beta chain for simplicity. The beta chain is made by this enzyme coming along and first picking one of the green guys and combining it with one of the purple guys. That's column C, column B and column C being combined together, snipping out the intervening DNA, and then that combined thing is then combined with one of the red guys. All right, so there's two, and the intervening DNA is snipped out. So that what you get when you're all done is something like this. Here's one from column A, one from column B, one from column C. So there's a certain amount of diversity that that can generate because there are in fact quite a few choices in these columns. Not infinitely many, but in the many tens. So sort of at that level of 10 to the 3, you have some diversity available. But that's not all, as they say on late night TV. At the same time as these things are being glued together, another enzyme basically chomps away at the faces that are joined together, and yet another enzyme randomly inserts. Okay, what is chomped away are bases, and what is inserted are bases in a random way. Now it's when you get to this point that you really start getting some diversity. So when you're all done, you have a combination of stuff that was originally in the genome, but selected and combined and slightly randomized. So let's see. This editing of the DNA to create a final gene that's then going to be expressed as a protein that's going to be this part of the receptor that goes onto the surface of the cell is random. That is to say, it's stochastic. For any given type of event, there are many possible outcomes, like there are many possible choices I could have made of these guys, the V genes, and you choose one of them with one probability, the other one with a different probability. You insert different numbers of bases in this insertion stage. Again, you could insert none, or you could insert one base, or you could insert 10 bases with different probabilities, and we'll come to that eventually. And that leads to, well, so there's a randomness in the whole system that's important. That's how the diversity is in fact generated. What's going on here? Why? Somehow this thing has gotten completely disorganized. All right, the output, let's see if this is going to, yeah, somehow we're now back in the right order. I have no idea what happened. Okay, so the output of this rearrangement can be observed by high-throughput sequencing technology. Namely, you can take a blood sample, you can just throw away everything except the immune cells, you can extract the DNA, and then it turns out that even though the whole point is that all of the genes that are, the gene that is of interest to me is essentially different in each cell, nonetheless there are some anchor points that are the same. So I can use the magic of PCR, polymerase chain reaction, essentially to, out of this large collection of DNA, to essentially amplify that part of the DNA that corresponds to this reassembled sequence for the surface receptor. And what emerges then is something like, if you do using today's technology, from a typical diagnostic blood sample, hundreds of thousands, if not millions, of sequences of the rearranged surface receptor sequence for these guys. The number of unique sequences will be somewhat less because you have many, sometimes multiple copies. Multiple cells have the same sequence because they have expanded. They've multiplied in your blood for one reason or another. Okay, so in the first instance, I'm not going to go through all of this in too much detail, but the thing that's of interest to us is to try to figure out, well, what are the statistical rules for, so to say, generating these recombined genes for the surface receptor. The best way to look at that, turns out, is to exploit the following fact. Since I'm doing stuff, damn it, at random, the likelihood that I'm going to and I'm picking slides more or less at random, The likelihood that in making random insertions, deletions, and all the rest of that, that I will have left the whole sequence in frame, as they say. That is to say, the total length is between anchor points that have to be there is some multiple of 3, so that you actually have something that properly translates. The likelihood of that is not 1. It's not even a third. It's somewhat less than a third. So there's some fraction of all of these sequences that will correspond to a sequence that got recombined all right, but it doesn't make a protein. It hits a stop codon somewhere in the middle, or this downstream something that should have been an alanine is a tryptophan or something like that, and it just doesn't work. So if you focus on those so-called non-productive sequences, then you get the following benefit. Clearly that cell didn't, that recombination didn't work. But why did you see it at all? Well, it's because when something doesn't work the first time you try, frequently the cell tries again. And if it worked the second time, then this first try, which was a mistake and just doesn't get expressed as a protein, just goes along as a fossil, all right? And no selective effect. In other words, once these guys get out into the blood and start working, some survive, some multiply, some don't, and there are, selection for function comes into play. And if we look at the so-called non-productive sequences, then no further selection for function is applied to them, and you have a direct record of what happened when the recombination took place. So that's the goal. Here, I think I'll skip over this. I was just trying to show you what, these proteins are long amino acid chains before they fold, and in these sequencing experiments, you don't sequence the whole protein. You could, but you don't. And you sequence a subset of it, and the thing to note is that it's typically 120, 130 base pairs for convenience, but there's some anchor residues, and this variability that I was talking about due to insertions and deletions is largely in something which they call the complementarity defining region sub 3. It makes a loop that sticks out from this protein, and it's sort of floating around in free space, and it's what is largely responsible for the specificity of recognizing a pathogen or whatever. And and that's the thing that we will, that's what's most interesting for the specificity and diversity of that specificity, and it sits in the middle of this region that the sequencers have decided very cleverly to sequence. Okay, now, here's a little bit of thought, analytic reasoning. Many scenarios for producing a particular sequence, there are many scenarios that will yield the same sequence, okay? And here's a cartoon in which there's some observed read, 130 base pairs of sequence that have come out of the sequencing machine. And how did you get it? Well, you could have gotten it by choosing this V gene, this D gene, this J gene, and deleting away some of this guy, deleting away some of that guy, and inserting stuff. Well, if you think about it, it's clear that, well, you could have deleted away a little bit more, and you could have inserted just by accident exactly the right thing to come back to the same place, and so on and so on. So deciding how this got made is really a matter of assigning probabilities to different, how shall I put it, events. So what are the events that create one of these recombined genes? Well, there are several in number, but they're not infinite in number. First, you have to make choices about which of these modules, these dishes in the 3 columns you chose. So there's some choice of V, D, or J gene. Having chosen a V gene, you have to decide, well, how many bases are you going to delete away? And similar for the other choices. Having made all those choices, well, how many bases are you going to insert between things that you chose? And what sequence specifically are you going to insert? And so on. So the probability of a scenario, that is to say, I chose this V gene, I deleted 7 bases from it, et cetera, I inserted 13 bases between the V and the D, you can make various reasonable sounding assumptions about what are the independent events, right? So, you know, an independent event would be, possible independent event is, well, I chose a particular V gene and it doesn't matter what happens next, there's just some probability for that. There's some probability that I made so many insertions between D and J. You make a reasonable assumption for how the probability for the whole scenario is composed of sub-independent probability distributions, all right? And then the probability of generating a particular sequence is then the sum over all of the scenarios that could have given you that sequence of the probability of each scenario. So this is great, except that you have no idea what these probabilities are, right? So for choosing out of the 70 different V genes, well, it could be 1 over 70 for each one of them. That's what you would start with, but maybe that's not right. Maybe it's 10 times more probable to have one of them than another, and so on. And there are good reasons why that might be the case. All right, so this is where the action really is. I'm going to call, I have a symbol called P-gen of sigma. It's the net probability that this particular read that we've seen will be produced in a single stem cell event. It's kind of the one-shot probability. I have a slot machine. I pull that slot machine to make a new recombined gene. It can make 1 of 10 to the 14 different outcomes. And if I tell you which outcome I'm looking at, I would like to know what's the probability that that one outcome came up. But we'll see that that's a useful thing to know. Okay. Okay, so we have a problem. We know what the outcomes are. We have these sequences that we found by putting blood into my sequencing machine. And we have a plausible model for the underlying kind of probability distribution for recombination events. And I would really like to know what's the answer. What are the actual probabilities for making, choosing the different genes, inserting different numbers of bases between pairs of genes and all the pairs of modules and the like. So this turns out to be and of course, the fundamental unit is if I have some guess for the probability distributions that go into this picture, that is to say, if I have some initial guess for, this P of V, or this P of DJ insertions, and so on, then I have a probability to be assigned to any given read. And then there's some likelihood of the whole thing, which is the product of all of those probabilities over the million different reads that I actually saw. And in principle, you could say, all I have to do is to mess around and try different values for these functions, and eventually I'll find something that maximizes the likelihood of the thing that I saw. And this sounds, well, this is a thing that you wouldn't want to do by hand or in your head, but you can do it on a computer, and there are what in the end are well-known statistical methodologies for doing this. Basically, you make a guess, you find the, let's see, I think I briefly say what it is. You perform an iterative procedure whose details I will not tell you about to find at least a local maximum, all right? And it turns out that in all of these cases, for reasons that are not too clear, the local maximum is quickly found and it's unique. It could have been that there were a gazillion different local maxima and you would then be kind of stuck, but in fact you always seem to converge on something that's quite unique. As I say, we use these so-called non-productive sequence repertoires, sequences that in fact are fossils. They were a mistake at the beginning and they never get really expressed as proteins and therefore are not subject to being thrown away for some functional reason. Okay, we succeed in inferring these probability distributions for the different events that taken together make these the final output. Now, one thing that I would like to emphasize is that, if I take the sequences that are in me or the sequences that are in you, the sequences themselves tell you very little about the immunological state of an individual. So what's really informative is the probability distribution from which they're drawn, and the goal is to really understand that probability distribution and maybe finding that it's different in different people in different states of health or something like that. The other thing to say is that the ability to tag any given sequence with its, so to say, primitive probability of generation opens up new analysis options. For example, and I'll show this, you can determine whether the fact that, well, out of the millions of different sequences that two different people may have, there are a couple that actually match, they're the same. Does that mean anything, or is this just an example of the birthday problem? That is to say, it's a random event. So you can look at that. And there are other things that you can do. So the point that I find rather nice about this is that after going through this exercise, you've identified a hidden variable. The hidden variable is any sequence that you find in somebody's immune system. This is a sequence which has a hidden variable, which is the probability that it gets generated in one shot by the recombination mechanism. Okay, so in the following I'm going to give examples of all of this for T cells, B cells, mice, and men, just for the heck of it. Okay, so here's an example of what comes out. So this is, as I said, there are quite a few. In human, there are some large number, I forget exactly what the precise number is, of entries in the first column in the menu, the so-called V genes. And here you see the result of the inference of the probability distribution of choices of the different V genes, and you can see it's all over the map. You might wonder why. We don't really know, but the point is that each of these segments, each of these possible things you can choose, has a little guard sequence to the right and a guard sequence to the left, and it's those sequences that are the signal for the enzyme that comes in and grabs it, and those sequences get mutated and they have some variation. And in addition, the different V genes are actually quite some distance apart, so there might even be some mechanical effect, but this is very hard to say. It's just a fact. The probability of using different such things is quite variable, and these error bars were variants across 7 human subjects. So Harlan Robbins, who was an ex-string theorist, I should point out, who got into this game really to do it at the experimental level, basically, and I got this data from him, basically took everybody in his lab and took 4 cc's of blood and did the analysis. Another thing that's kind of amusing to look at is that, okay, this is the physical layout of the V, the red, the D, the purple, and the J, the green, and things get recombined from right to left. So you can see here that this D gene can only recombine with this, and it turns out that in this case there are 2 D genes and the J genes are broken up into 2 subgroups. Sometime, somewhere, there was a gene duplication event. So this D can only recombine with guys to his right. This D can recombine with either of the 2 groups. And that's what's shown up here when we looked at the probability of combining a D, finding a D connected to a given J. This is 100%, no, this is 0%, and this is 100%. In other words, the statistical analysis completely confirms this topology, if you will, of the way the recombination happens. Let's look at one other thing before we go too far off the rails. What's more interesting, for reasons that'll become clear, is what happens in these insertions, because that's where most of the diversity comes from. So, you know, one of the factors in this probability distribution was probability of inserting, you know, 1, 0, 1, 2, etc. bases between the pairs of genes that have been brought together. And so this is a plot of that. It's a little bit messy because I didn't construct for you a clean new plot. This has got too much information on it, but you can see that, well, there's some probability of insertions, there's some probability of 0, the probability rises to a maximum at 4, and then it falls off in some vaguely exponential fashion. There are some error bars on here. These error bars are simply variance across individuals, so you can see it's pretty stable from individual to individual, and, well, you know, the mean number of insertions is somewhere, I don't know, somewhere between 5 and 10. Here's some information on the randomness of the bases that get inserted, and this is simply meant to say that Well, there is some preference for different kinds of base, and it's pretty stable from individual to individual. And basically, actually, it's not just single bases, it's dinucleotides that really describe the statistics of what gets inserted. Okay. Why is there a peak at 4 insertions? Don't really know. There could be some mechanistic understanding of this. But in any case, the point is, it's possible to winkle out what is the distribution function for length of insertions. And there are 2 places where you get to insert between the VD and the DJ, and those 2 distributions fall right on top of one another. Okay, so for the physicists, what's kind of interesting is to look at the quantifying the diversity of what comes out of this. So this somewhat busy plot is meant to tell you all about that. So I have a probability distribution for the events that come together to make this recombined gene, and I can give you the entropy of that distribution because I have explicit functions. Probability of V is some numbers going from 0 to 20 or whatever, and I can combine them all together and compute the entropy. So the entropy of recombination events is 52 bits. It's a lot, right? I mean, this is, what is it? It's 2 to the 10 is 1,000. It's like, well, it's 10 to the 15 or something like that. That is not quite the same thing as the entropy of nucleotide sequences, because as I said, there are multiple ways that you can recombine to get the same sequence, and you can actually do a reasonable estimate of that. And it turns out that, so to say, convergent recombination actually is responsible for 5 bits. So the total nucleotide sequence diversity is 47 bits. I won't tell you how you do this, but it's pretty simple. Okay, so the graphic is meant to show that, well, this overall diversity, or this entropy, is made up of different pieces. You know, there's some diversity that's associated with the choice of different V, D, and J genes. That's what I talked about at the very beginning. There's combinatorial diversity. There's not a heck of a lot there. Ditto for these deletions that get made. Most of the entropy is coming from insertions, and you know, it's relatively equally divided between the two regions, VD and DJ. So these are numbers that are worth keeping in mind. And the actual potentially unique T cell receptor beta chains that this corresponds to is something like 10 to the 14. In your body, you don't have anywhere near 10 to the 14 distinct sequences of this particular protein. You've got maybe 10 to the 7 or something like that. Why do you need this overkill? We have no idea. And more comes on. So, okay, so this is all, the question is whether this is just mad imaginings of a theoretical physicist, or does this have any connection with reality? Well, you can have a look at this question of overlap between individuals and think about does the occurrence of the same sequence in 2 different individuals, does it have any other meaning than chance? So you can answer that question now that you know, here's a sequence. The probability that it got generated was this number. Okay. And I can take, since we had hundreds of thousands of sequences from 7 different individuals, there were many opportunities for, so to say, coincidence between a sequence and 2 different people. And we could calculate what is the probability of overlap, what's the probability that in any pair of individuals there could be single coincident sequences, multiple pairs, and so on. So you can get an actual, that is to say, the actual numbers of shared sequences between different pairs of individuals, and you see the more pairs of individuals you have, the more shared sequences you could have, and so there's a prediction, that's the blue line, from the actual probabilities that we have inferred, and then there's the data, okay, the red dots. So Poisson's statistics seems to cover very nicely what's going on with shared sequences. The part that another way of plotting the same thing, which I like a lot, is you can take the sequences from any given individual. Each one is tagged with a number, which was its one-shot probability of being generated. Wow, okay, very good. Well, I'm almost done. So this blue curve is a plot on, I don't know, call it semi-log, of the sequences, the observed distribution of probabilities of generation in an actual repertoire taken from an individual. The red is take all of those sequences which are shared. Now, by any, obviously if this assignment of probabilities is anywhere near correct, the shared sequences must be ones that had a high probability of being generated in the first place, and that's what this red curve is telling you. Okay? All right, so let's see. What I want to tell you in the couple of moments left is, all right, that's what happens for human beings. Human beings have not only T cells, they have B cells. So the T cells are really good because they are in the business of detecting proteins that are inside your cells that shouldn't be there. And I won't go into the mechanism, but that's their role. B cells basically are surveying your tissues for direct presence of some molecule that shouldn't be there. So B cells are different, and B cells and T cells have certain cooperative arrangements, but you can go through the same exercise for the B cells. Easy enough, easy for me to say that it's easy. You can get the sequences for B cells from human subjects, and here I plot, just for reference, the insertion distribution. The reason why I plot insertions is that most of the diversity comes from them, and maybe it's not totally clear from this, but if you do a head-to-head comparison of the T cell case with the B cell case, you will see that this really has a much broader distribution. The average number of insertions is much bigger. The result is a giant increase in entropy of potential B cell sequences. Why is this needed? It's factors. The nucleotide sequence diversity for the B cells is 74 bits. It's 25 bits more than the diversity of the T cells. We don't know why B cells need to be so much more diverse, but they are. And let me just back into one more thing. Are human beings really different from other organisms? Well, it's easy enough to get similar data from mouse. And the reason why I put up, and then we've done that, and we perform a similar sort of analysis. And you see one thing that's really quite surprising and interesting, and I will at least mention it. So mice are a dime a dozen, and it is possible to, and people have laboratories where they have large numbers of mice available for study. So you can do time series experiments. So there's two time dependence issues that are of relevance. One is, well, what happens when you're infected or vaccinated? You'd like to know what happens to the immune system over time. And the other thing is, what happens, well, there are three things. What happens as you're being gestated and born, and what happens as you age? We all know that as we age, something unpleasant happens to our immune system. This is a big subject, but what I've got here is some information about what happens when you are first being an embryo and then being born. There's quite a dramatic change in the distribution of these insertions in the mouse as you go from being an embryo to being a few days old to being a few weeks old. There's some dynamics going on, and we begin to understand a fair amount about it. But the other thing to note is that as a result of that, what you can see is in the embryo, the mouse has almost no insertions in his T cell proteins. And as he grows up, he starts to get more. But even as an adult mouse, he really doesn't have anywhere near the diversity of the human system. I don't know if this is clear enough to see. So remember, for the T cells in the human, the overall nucleotide sequence diversity was of order 48 bits or something like that. For the mature mouse, it's like 30 bits, so it's 18, so almost 1,000,000 times less diverse. And the embryonic mouse really is just hardly diverse at all. And it's kind of interesting because the mouse, you would think, lives in a pretty crappy environment, sort of dirtier than ours. You'd think he'd have to defend against more stuff than we do, but somehow he gets away with a much less diverse immune system. Okay, let me just pass on, since I'm being wigwagged, to, so, problem that I've not been able to tell you about, I've never been able to get this across in any kind of venue yet, is what happens when a T cell actually gets produced and goes out into the world and gets selected for function. We actually learned a fair amount about that, but I won't tell you about it. So what have we learned? What remains to do? So we've learned that repertoires from different individuals have almost no sequence overlap, and the generative models that govern how their diversity is generated are very similar from individual to individual. The mouse is very similar to the human. Insertions are fewer, diversity is much less, and there's some structural business which I won't go into. We think that the ability to tag each sequence with its one-shot probability of generation is conceptually important and will allow us to do lots of interesting things. Now, so there's a big problem, which is, so to say, where this is all going. And this is my last 30 seconds. In order to discuss immune response to antigen, we need sequence data on both the immune system and the infecting pathogen as they co-evolve, right? So think about HIV. HIV is a virus that unfortunately infects the immune system, and the virus is evolving and the immune system is doing its best. And evolving is a word you have to apply to the immune system with a certain amount of care and caution. But there are, so to say, two different systems that are interacting strongly, let's put it that way. So we have a statistical, so basically I've been talking about understanding something about the static statistical properties of one side of this war. So the immune system is at war with viruses and bacteria. The viruses and bacteria evolve. The immune system has the ability to respond to evolving threats. And there's a really interesting theoretical problem which first has to be illuminated by some data. You need some data to get a first crack at this. And interestingly and happily, I would say, the data that will allow us to begin to develop models to understand, or at least quantify, how the two sides of this competition interact with one another in a statistical mechanical fashion is coming down the pike. So really interesting challenge to be taken up. Okay, so quantitative understanding of big data of the kind we seek to achieve will be pleasing to have and might even have some value for medicine someday. So it's science, and maybe even applicable science eventually. And happy birthday.